So here with my pen, that decides not to work. So we are going to talk about the secondary lymphoid tissues, right? But before we get to the anatomy and physiology of the secondary lymphoid tissues, I think I need a new computer. Okay, here we go. Okay, between alpha 1 and alpha 2, okay? But you look here at this groove of antigen binding site. It looks different than the groove that you have seen on MHC1. So this groove here looks more like the groove that you see on this MHC class 2 molecule. It's a deeper groove, okay? So you can put bigger things in there. But remember, this CD1 family is not going to bind proteins. For the proteins, we already have MHC1 and MHC2. So these guys here are going to bind something else other than proteins. Most commonly is going to bind some kind of lipid, okay? So what's going on here? It kind of looks like MHC class 1, but it is behaving like MHC class 2, and it's not even presenting protein. It's going to present something else. Okay, so where is it that you're going to find this CD1 family of antigen-presenting molecules? You're going to find them on antigen-presenting cells. Okay, so your antigen-presenting cells are going to have also some of those CD1 family molecules. Now I go here. So here is your most important antigen-presenting cell. So here is depicting your dendritic cell, right? And you have all these different members of your CD1 family. So you call those your unconventional antigen-presenting molecules. Right, so you have them, as you see here, in the human and the mouse. So if we're only really interested in human, right? So human, the mouse will have, you know, some of those that we have, but then we have some of the CD1 family members that mice don't have, and that's a bit inconvenient because you cannot really do a lot of research on these molecules using the mouse animal model, at least, right? So that's just like research practical issues, right? So what is happening here? Those MHC molecules, they are going to interact with the T cell receptor, right? But then you're going to have some special T cell receptors that can recognize things that are not proteins, right? And they could be able to interact with those CD1 molecules. So the production of those T cell receptors is a little bit different than the genetic manipulation that we have seen before for the T cell receptors that are going to recognize proteins. You know? So basically, you are going to have a T cell receptor that does not really have a lot of variation, as we have seen before, that happens with the regular T cell receptor. So we call those semi-invariant T cell receptors, right? So basically, they're going to be behaving a little bit like innate type of detection, like those PRRs, right? Pattern recognition receptors type, okay? Because they are going to identify pieces of not proteins, right, that basically have more or less the same shape. So if you look at lipids, right, you remember if you took biochemistry or cell bio, right, you remember what the lipids look like. They're like chains of carbohydrates, right? And also they're going to be able to detect some sugars, and those are also, you know, made up of carbon things, right? So they don't really change much, but they have certain patterns that are present in pathogens, right? And when you chop up those pathogen pieces, they can be detected by this family of CD1 molecules. Like there are many CD1 molecule families, right? So you see here, for example, CD1A, B, C, and this guy that is not a CD1 is called MR1, but is part of the CD1 family. So it gets complicated, but we're gonna just keep it very, very simple. So what are the T cells that can detect CD1 family members? You have here your conventional alpha beta T cell, but most importantly, you have your gamma delta T cells that can detect the CD1, okay? Now, let me see here. Okay, so here are examples of the types of things that can be presented on the CD1 and can be detected by those T cells or T lymphocytes type. Okay, so I see first what are the T lymphocytes that can detect and can interact with your CD1 family. So here you have your natural killer cells, okay, the two types of natural killer cells, so type 1 and type 2. Okay. What they are going to do, they're going to use the T cell receptor that they have, or semi-invariant T cell receptors, right? Because remember, natural killer cells are part of your innate immune system, right? And those are going to be able to detect sugars and some lipids that are presented on CD1 molecules. Okay, so you see here your natural killer cells detecting these sugars that are derived from mainly from bacteria. Okay, and then you have here your special type of T cells that have T cell receptors that are meant to detect CD. And those cells are not natural killer cells. Okay? So they have this CD1 on the surface, and they are going to detect mainly sugars, again, okay, sugars, or fats. Where are the lipids here? Okay, let me see. Okay, the main idea here is that you do have these specialized T cells that are able to detect sugars and lipids, okay, presented by those CD1 molecules. Okay. No, you do not have to know details about these things here. Just remember that CD1 is going to present either lipids or sugars to your specialized T cell receptor. Okay. All right. So how is it that you're going to get a piece of lipid on your CD1 molecule? Okay. The idea here is, again, that your immune system can detect most foreign things that get to your body, right? So a lot of the antigens, not just the proteins, a lot of antigens can be recognized. Like, except some antigens that are a bit strange, mainly because they were not part of nature, or they were not supposed to be in your body in the first place. Like, for example, here, we say plastics don't really get detected by the system as far as we know, and things are always changing in immunology as we learn more about it, right? And they're saying metal, also like metal implants, do not get detected by this system. And the question here is why? Yeah. No PAMPs. No, if you look at plastic, at nature of this of plastics, have thousands of different types of plastics, and usually, you know, they are polymers, so they have repeating units. So you have PAMPs there. Yeah.
Could it be like the binding interactions between the CD1 receptor and the glue and those compounds?
Binding interaction, you're getting there, but the whole idea is still missing on that link, but you're on the right track. Yeah.
And like, is it just because maybe our immune system isn't strong enough to break down toxic metal?
Exactly. Yeah. You can't break it down. It's not strong enough. It's just that you don't have the enzymes to break them down. Because it's strong, it's just not going to be able to break it. Okay. So If you get a piece of plastic and you want to process it and chop it up and put it on the presenting molecule, your body is not really able to do that as far as we know. But things are always changing because we're always learning new things about the immune system, right? Especially when you start dealing with those blessed nanoplastics, that they are so tiny that I do think that they get presented somewhere. Okay, they might get presented in this system. All right. So what is happening here with the lipids? When you have lipids that are, in the extracellular fluid, it is eventually going to get endocytosed by those presenting cells, right? And when you go through the breaking down process of the lipids, you have enzymes that break them down, and those are the lipases. Remember, enzymes that do the job, get the A's at the end, those are breaking lipids, so we call them lipases. So those get chopped up. You have here the phagolysosome that is getting together with a vesicle that has your CD1 family member, and then your piece of lipid gets put on the surface and presented on the cell membrane, right? So here, when you have this lipid on the CD1, you're going to have either your alpha beta T cell or your gamma delta T cell or a natural killer cell that is specialized to go there and interact with this presenting molecule. Okay, so you can then start mounting your adaptive immune response to that molecule. So I put here, yeah, vesicles join, happy party at the cell surface, then no way. Okay, finally here is your question that I promised you from last time. So here, come on, today. Okay. Wait, it's come, yes, it's alive. Okay. Okay, so here, select, let's see if it works now. Yes, I think it's working. Okay, so the answer here is, yeah.
Going back just from the last one, but last class.
No, we're good, we're good. All right. So here we go into stuff that is happening at your lymphoid tissues, but then before we really get to the structure, we really have to wrap up on something else that is happening here with some very important cell, like your B cells activation, right? So we looked at before how the other lymphocyte, how your T cell gets activated, right? So now we're going to look a little bit more on this mechanism for the B cell activation. That is going to happen at your secondary lymphoid tissues, right? Same thing, the T cells are getting activated at the secondary lymphoid tissues as well, okay? So what we're going to look here, we're going to look at what is happening at those germinal centers in the secondary lymphoid tissues, and we are going to look at actions of this important follicular helper T cell. Actually, going to look more at a follicular dendritic cell that is happening, activating B cells at the germinal centers. Okay, so let's see what's going on here. All right, so just to recap, what are your secondary lymphoid tissues and what are the cells that you find there, right? So secondary lymphoid tissues are all the tissues, but not your thymus and your bone marrow. Everything else involved here with your lymphatic system is considered secondary lymphoid tissues. So the cells that you're going to find there, you're going to find all sorts of T cells and your B cells. You're going to find your differentiated B cells, that are your plasma cells, right? Then you're going to find your very, very important link between innate and immune system, that is your dendritic cell. But besides your typical dendritic cell, you're also going to find this guy here, the follicular dendritic cell. Okay, this guy is going to be really, really important at activating further differentiation of your experienced B cell. Okay, we're going to see how that works. So besides that, you are going to find your macrophages, right? The surveilling macrophages over there. Okay. All right. So recap here, what is happening with this journey of your B cell? Remember, the B cell gets to develop in the bone marrow, right? And then in the bone marrow, your B cell undergoes that process of negative selection, right? Remember what the negative selection is doing in the bone marrow? You have some stromal cells that are presenting self particles, self antigens. If your IgM of your B cell is able to interact with those self antigens, then the cell is going to die. So that's your negative selection, right? Now, okay, so let's go by the thing what it's doing here. After the cell leaves the bone marrow and goes to the periphery, right? Now, at the periphery is when you are going to present your antigens that are foreign for activation of that B cell, right? So remember that you have several ways of activating your B cell, and one important thing that is capable of activating the B cell is the presence of those antigens on surface of a pathogen. That means you have clustered antigen presentation, and that means you can activate more than one of your B cell receptor at the same time. Remember that to activate your B cell, you need at least 2 signals, right? So if you have only one signal that binds to one of those B cell receptors, you're not going to activate your B cell. And this is really, really important for tolerance, right? So after you activate your B cell, you start producing antibodies. Okay. This quick recap. Okay. So here, the idea of your central tolerance, right, is happening in the bone marrow. Okay. And then when you come out to the periphery, you are also going to further select your B cell for non-reactive self B cells. Okay. The idea here is that in the bone marrow, the B cell is not really exposed to all of the possible self-antigens. Right? It's a lot less self-antigen presented to the B cell in comparison to the self-antigens that get presented to the T cell in the thymus. Because the thymus, remember, it has that very, very important transcription factor. Remember ARI transcription factor? The Swiss knife kind of transcription factor? So that is not present in the bone marrow. That is only present in the thymus. So the T cells really get a lot of schooling on self-antigens, but the bone marrow is kind of lacking on seeing self-antigens. So after the B cell comes out of the bone marrow, it has to go through some extra maturation steps in order for it not to attack self. Okay, and that's going to happen really at the spleen. Mainly it's going to happen at the spleen. Okay, so when this thing happens when negative selection happens in the bone marrow, you call that central tolerance. Okay, when it goes to the periphery and you do further refining of the tolerance, you call that peripheral tolerance. Okay, if you still have B cells that can sneak past those selection of central and peripheral tolerance or negative selection, then you're going to have problems. That's when you start having autoimmune diseases. Okay? Examples of autoimmune diseases that are due to this type of sneaking past your negative selections are saying here. So you have like rheumatoid arthritis and also systemic lupus erythematosus. That those are types of diseases that are due to self-antibodies. All right. So here, a bit more on that B cell negative selection, right? So we've seen this. We've seen this, that multivalent self-antigen is going to trigger this cell to die. That's what we've seen so far. The parts that I didn't mention, they're right here. Okay, so let's pay attention here. Still in the bone marrow, if you have little antigens that are self-antigens that are floating in the extracellular fluid space, okay? And those soluble antigens are capable of interacting with your IgM, then this is not going to trigger death of your B cell. Okay, so if you have soluble antigens, they are you're going to give one signal to your IgM, and that is not going to trigger apoptosis. And I know this this picture is a bit misleading because it's showing you the antigen binding to 2 antibodies here. That's not very accurate. Okay, so this would be one signal, not 2 signals. Okay, so make sure that you make a note of that. Soluble self-antigens do not trigger B cell activation. And soluble self-antigens in the bone marrow, they are not going to trigger apoptosis of the B cell that recognizes them. Okay? Okay. All right, so 2 things that are possible here with self-soluble antigens, right? You are going to have a B cell that is not dying, that is not activated, but then at the same time, if you only send one signal to the cell, this is how you get energy that is tolerance induced tolerance, right? So the cells that are able to come out and not die, they are not going to be reactive. They're going to just be floating around for some time, like zombie cells, and eventually they're going to die. Okay? All right. So same type of idea goes with soluble and not very binding self-antigens. Okay? You're going to produce anergic. Oh, here's the zombie-like cell. Okay, anergic zombie-like cells. Important for tolerance mechanism. Okay. So here, when you have your bone marrow that is really undergoing that negative selection, you really have 3 fates for your B cells. They can either die, or they can become zombies, or if they have the option, they could try to reshuffle that light chain and edit the receptor. Okay, to try to become useful in some other way. Okay. How many cells die during this process? Well, basically most of them. Most of the developing B cells are going to be eliminated by negative selection. Look at this. About 90% of the B cells that are produced are killed. Okay? Because of self-reactivity. Okay. Everybody cool with that? Okay, so now what happens to that B cell that left the bone marrow and goes to the periphery? So here they're going to get to your secondary lymphoid organ. Mainly, you're going to get to the spleen. So the cell is going to get to the spleen first. Okay. Now, when the cell goes to the spleen, it is going to get a signal to activate this production of this maintenance molecule that we call the maintenance molecule here. We call it the B cell activating factor. It's a long name, okay? So take a deep breath here. B cell activating factor belonging to TNF family receptor. Yeah, that's the name of the molecule. To make life simpler, we call it the BAFF. BAFF, or you can think about it like the B cell best friend, okay, friend forever, which makes sense. B cell friends forever. Why? Because if you do not activate the expression of this BAF, of this B cell friend forever, then the B cell is not going to survive for a long time. Okay, so in order for the cell to survive, it needs to have this BAF, this best friend on the surface. Okay, so maintenance of the cell survival has to do with the presence of this BAF on the surface. Okay, if the cell doesn't get that, then the cell is going to die. Okay, it's going to undergo apoptosis. All right. This is going to happen at the spleen. Okay. So what's going to happen there? Cell signal for survival. You're going to get also signal for further differentiation and further maturation of your B cell. All right. So we talk about this part over here. Okay, maintaining your B cell, and then after the cell is kept alive, then it is able to see antigen and differentiate. Okay. So what is happening here at the spleen? In this session over here, it is all happening at this marginal zone. Okay, it's not really happening at the B cell zone. It's happening at this marginal zone. And basically, when you get activation of the B cell at the marginal zone, you're usually going to have activation by T independent processes. Okay, so that's T independent. Right? So at this marginal area, it didn't really encounter the T cell, and you're just going to get those activations that allow the B cell to produce antibodies, but those are going to be IgM. They're not going to be very high affinity, right? And the cell, after a while, it's going to die. Okay, it doesn't last, it doesn't stay for a long time. Okay? So you can imagine that if you are using those pattern recognition receptors that are binding to sugars, right, that are on those bacterial polysaccharides, that's the type of T-independent mechanism of activation of your B cell. And what end up happening is that, yeah, you can produce some antibodies, but the cells, it didn't really produce memory cells, and those cells that are producing those antibodies, they're eventually going to die, okay? And you're not going to have a very long-term immune protection against that pathogen, okay? Now, think about it. If you see, if you get your B cell activated only by seeing the pathogen like that, without T cell help, then your immunity is not going to be the greatest. Okay, so this is the principle for some types of bacterial vaccines that we get, or some toxin vaccines that we get. So we don't have long-term memory cells development, and eventually you have to be taking boosters of the vaccine, because it is really activating your B cell by T-independent pathways. Okay. All right. So this is T-independent activation. Still, you need expression, continuous expression of your best friend for the cell to survive in that session. Okay. Now let's say that inside of your spleen, let's say that now the B cell went to a follicle, right, to the B cell zone. And at the B cell zone, you are able to activate it by T cell, T-dependent activation. Okay, so in the B cell zone, you can get your T-dependent activation. So this is when you are able to form a long-term antibody, a long-term B cell memory. And you're also going to be able to do affinity maturation and further development of your B cell. Okay, it's going to get clearer in a bit, okay? Okay. All right, so The cells that don't readily upregulate the best friend, okay, they hang around for some time and they are not totally useless. They are still able to produce not only IgM, but they can also produce some IgA antibodies, okay? And this is really important because IgAs are the ones that are going to get to your mucus and it's going to be secreted, right? It's going to be secreted in the breast milk and in salivary glands and the sweat, and most importantly in your GI mucous membranes, okay? So you want to have those antibodies being able to do their job, okay? And this is a really interesting thing that happens here. Look, if you have a person that doesn't produce a lot of this B best friend receptor, okay, you're going to have a problem of producing antibody class switching, right? Because without continuous stimulation to be kept alive, the cell is not going to be able to live long enough to interact with T cells and do class switching, okay? So without this best friend of the B cells, you still can produce your IgA, because that's what happens when you have the T-independent type of activation, okay? But you don't do class switching, so you cannot produce IgG, and also, amazingly, you get low level of IgM production too. So basically, if you see a person that has defect in this gene, you see normal levels of IgA production, but you don't really see normal levels of the other isotypes. Okay, they didn't even mention IgE here, but you can imagine it's also low IgE. Okay, so normal IgA levels, but low levels of the other antibodies. This is indicative that there was no problem of B cell developing in the bone marrow because you have B cells, right? There is no problem of activating the B cells because you're getting antibodies, okay? So there must be a problem on maintaining the B cell alive so that it can produce the other types of antibodies, mainly your T-dependent class switch of antibodies. Okay, so that will be a telltale sign that your cell is not living long enough in the periphery to be able to undergo the class switching and further differentiation. Okay.
Can that be treated?
It could be treated if you're thinking about genetic engineering or stem cell therapy, yeah. Or you could try to introduce a virus in the bone marrow to correct the defect of the expression of the gene. Yeah, these types of genetic problems theoretically can be treated, but practically it's a different story. Yeah, we can find cures for a lot of things that are genetics, but you know, in practicalities, yeah, good luck with that. And then good luck to get your insurance company to approve this treatment. But yeah, in theory, you can treat it, yeah. All right, so problems with expression of your best friend here. First problem is that you're not gonna have a proper secondary immune system functions, right? You can have IgA, but the other guys are not quite there, right? And there could be also other problems that could arise. In case you start producing too much BAFF, okay, that could be a problem too. Because if you start producing a lot of cells that hang around for a very long time and never get turned over, now you can also develop some types of autoimmune diseases. So the example that is given here is Schrodinger's. I can never pronounce this thing. Sjogren's syndrome. Okay, it's a problem of high levels of antibodies, and is also considered autoimmune disease. All right. So here, coming back to your secondary lymphoid tissue, right? And what is happening here for the parts that are important for activation of those B cells, okay? So let's see what is happening here. Your B cells, when they get to your, this here is a lymph node, okay? So when you get to your lymph node, okay, you are going to come through the afferent lymphatic, or you could also go through the high endothelial venous, right? So B cell is going to get there. So remember, you're going to have like two types of B cells. One type of B cell is going to hang around in a place that is more like a marginal zone and not really in the B cell zone. So B cell zone is the follicle, okay? So here it is at the okay, now that came out of my mind. Marginal zone is more to the outside. It's not at the medulla. It's more to the cortex. That's the word. It's close to the cortex. This is a cortex area. Okay. So B cells, some B cells, most of the B cells actually is going to go to the follicle. You call those your follicular B cells. Okay, so that'd be 2 cells. Okay. But then some of the B cells, you're going to stay around the marginal zone, and then you call them marginal zone B cells. Okay. All right, so looking here at your follicular B cells, this is the type of B cell that is going to be able to interact with your T cell, okay, and is also going to be receiving those antigens that are going to come in one of 2 ways. So one way that the antigens can come to that B cell at the follicle is that they can go through these little conduits that are formed at the cortex, and if they're small enough, they'll go through and they'll get to the follicle, and then the B cell will be able to phagocytose them and put it on MHC class 2, right? And another way is that you can have your antigen presenting cell, in this case here's a macrophage, but you could have your dendritic cell that is going to bring this antigen to the follicle and present it to the B cell. Now remember that in order for the B cell to see antigen, it has to see it, the whole thing, it has to see it in 3D. So this antigen presenting cell is just going to pick it up very carefully. It picks up the antigen and it brings it, without destroying it, to the B cell and then gives it to the B cell. Okay, so this is a different type of presentation, right, in comparison to the T cell. All right, so you can either have it handed by your presenting cell, or you can just catch it if the antigen is small enough to go through the conduit, and you can catch it at the follicle. Okay. So this is what I'm saying, what I put it in writing here. Once your B cell sees the antigen, you know it's going to start dividing, right? Because you only have maybe 1 or 2 B cells that can see the particular antigen. So you're going to get that clonal selection, okay? Oh, here's your B cell bringing also, some antigens to be presented over there. Okay. So here, just to reiterate on what is happening with your B cell activation, remember that you need at least 2 signals. So here is your T-independent type of signaling, right? Here's a B cell receptor, here's a little bacteria. So you have a recognition of your antigen, and you have a second signal here, for example, that got activating the CR2 type of receptor. Okay, so 2 signals and you get proliferation. Remember, this is T independent, So you're not going to have that CD40 to CD40 ligand. That is only for the T cells. Okay. All right. Here's the other example of T-independent activation, right? T-like receptor. All right. So now B cell got activated. Here we are at the lymph node. We are here at your B cell zone, right? And now you are going to start to proliferate that cell. When the cell starts to divide, you call this B cell zone, you called it a primary follicle. Okay, so that's your primary follicle. The cell is starting to divide. Okay. And okay. All right, so the cell recognizes the antigen, starts to divide this primary follicle. After you divide a lot, you become a secondary follicle. This is just terms of, are you just seeing the antigen, or you just or are you proliferating a lot? All right, so important things to keep in mind about this lymph node. Remember, this is your afferent vessel, right? And here in the center, you have a medulla. And then at the sides, you're going to have the cortical area. So your B cell zones are in the cortical area, okay? All right, so what is happening here with this B cell activation and differentiation in the lymph node, okay? So the B cell gets there. This is all inside of the lymph node. This is all inside this cortical region, okay? So this is your follicle. This is where your B cell is going to be proliferating and differentiating, right? So here is this paracortical area. This is really the part where you see your T cells hanging around. So this area here that you have your germinal center is where the B cells are. But outside of this area is this peripheral area where your T cells like to hang around. But we are talking here really about your B cell type of differentiation, right? So B cell gets there. If it gets activation by your T cell, then it can form your primary follicle, a secondary follicle, and get activated into differentiating those antibodies, okay, antibody class switching, right? So B cell gets there, it is interacting with the T cell, it gets activated, it's going to form the germinal center, okay? At the germinal center, the B cell starts to divide. Some of these B cells are going to produce antibodies and hang around and start to secrete antibodies. Some other B cells, they're going to hang around at this germinal center, and they're going to do the class switching. They're going to, they got exposed, they're getting exposed to the T cell, the helper cell, and they're going to do class switching. But at the same time, they are going to hang around the germinal center, and they are going to interact with this other dendritic cell right there. This dendritic cell is a very specialized follicular dendritic cell. It does not come out of the follicle. It just stays there. The job of this follicular dendritic cell is to present antigens to the B cell receptors and stimulate affinity maturation of those B cell receptors. Okay, so this is all going to happen inside of the follicle. Okay, so what is happening here? So the dendritic cell, follicular dendritic cell, activated differentiation of the B cell receptor. Some of them are good, some of them are bad, like good affinity for antigen or worse affinity for the antigen. Okay, if you have production of B cell receptor that is lower affinity, then it's useless, so the cells are going to undergo apoptosis. Okay, if you have a better affinity maturation, then you're going to keep on working with that, that clone in order to produce better antibodies. Okay? All right. And if it happens that you produce a version of a B cell receptor that becomes autoreactive, then you're also going to kill that cell, okay? Because you don't want autoreactive B cells to develop out of there. Okay. Okay, so this is exactly what I just mentioned. Okay. Okay, so here, important function of those follicular dendritic cells. Okay, important function for interaction with your developing B cell. They are going to produce a couple of important cytokines. They're interleukin 6 and 15, and they are going to induce production of BAF. Okay, so BAF is the cell survival signal for your B cell. All right, so what is important to know here? All right, so here, reactions things that are importantly happening in your germinal center, right? You're going to have here your affinity maturation, you're going to have your class switching, and you're going to generate your plasma cells that have better antibodies. Besides that, you're also going to produce those very, very important memory B cells. Okay, so memory B cells are mainly going to be produced after all of these interactions that are going on at the germinal center. So in the germinal center, you produce those memory B cells. Another important thing that is happening at the germinal center is this interaction with this T cell over here. Okay, so you know this is a T cell, this is a helper T cell, it's helping the B cell, but this T cell here, it has a further classification that is called the follicular helper cell. So this is a follicular helper T cell. It stays in the follicle, and its job is to interact with this maturing B cell and to coordinate class switching of that maturing B cell. Yeah, I know there is a lot of stuff happening here, but it should all make sense. Okay, so if you start thinking on overall developing of the B cell, where the B cell has to go, how is it that the B cell is going to see antigen, okay? And once it sees the antigen, what is it that the B cell has to do in order to be effective? How is it that it's going to produce proper antibodies? How is it that it's going to get to produce memory cells to hang around, right? And how is it that you're going to get rid of cells that develop receptors that are not useful? Okay, all of this stuff is happening at the germinal center. Okay, what else is important here? All right, gets more complicated. Sorry, not my fault. Okay, subdivisions of the germinal center. Okay, this is the last part of the germinal center, and I'll tell you something. But these things get useful later on when you go to graduate school and you see these things. Okay, so important things to know at the germinal center. So this thing here is the germinal center, right? So the cell gets there, and look, there are two tones of blue here. One is darker and the other one is lighter. Well, that's because you have a subdivision of your germinal center. So thank goodness we call it the darker part, you call it the dark zone, and the light part, you call it the light zone. That's simple. All right. What is happening in the dark zone? You are going to have a B cell that has seen antigen, so it's not naive, that's an experienced B cell, and the B cell is going to proliferate a lot, okay? Besides that, you're also going to do some hypermutation of your B cell, okay? So you are going to get the B cell activated, and you're going to get the B cell to transform the B cell receptor. All that hypermutation that is happening at the B cell receptor is happening in the dark zone. How can you remember that? Well, replication or cell division or proliferation of the B cell is the kind of thing that the B cells like to do in the dark. So you can imagine those B cells are just creating babies of themselves, and usually it is in the dark, if that helps any kind of association in your minds, okay? Happening in the dark. What is happening in the light zone here? Okay. In the light zone, you are going to select what is useful and what is not useful. So if you have B cell receptors that have low affinity to the antigen or lower affinity than the original, you're going to kill the B cell. Okay. If you have B cells that are self-recognizing, you're also going to kill it in the light zone. Okay, so useless B cells are going to get killed there. All right. Besides those two zones, around this follicle, you have a little cover that you call it the mantle zone. Okay, so mantle zone is going to be covering this follicle like a blanket. Okay. In this mantle zone, you're going to find your plasma cell that is going to be shooting out antibodies. And also in this mantle zone, you are going to have your memory B cell that is going to show up there. Okay, so shooting antibodies and developing of your memory B cell. Okay, it's right there. Memory and plasma cell shooting antibodies in your mantle zone. All right. So here is just another picture showing these different zones, okay, at your germinal center. This one I took from Lecturio, okay. So you just have here the different things that are happening at these different zones, okay. Dark zone here, proliferation. Light zone here is stimulation and affinity maturation. And then it comes to the mantle, and you're going to have the plasma cells and your memory cells. They're going to be around there. All right. What is the use of our memory cells? Well, the memory cells, they are not naive, so they do not need 2 signals to become active, because they already had those before. So the memory cells, they only need 1 signal to get activated, okay? That's why they're so important, because this 1 signal that they need, what do you think it is? Huh? The antigen, right? Antigen. The antigen. When these memory B cells see the antigen, it is that 1 signal that it needs to get activated. And this is how you get a very fast and strong secondary immune response. Okay, it's because of those memory B cells that they only need to see the antigen. Okay. All right. So once you activate your memory cell, what does it do? Well, it also goes to the follicle to see if it can become better. Okay. So you already have memory cells. You're always going to have some memory cells. But now once it gets activated again, it goes to the follicle and it says, well, do you think I can make even a better antigen receptor? Can I have a better B cell receptor? Can I be stronger? Can I be more effective? So it's still going to try to improve the memory B cell. And this is why it is important to get booster shots of vaccines, okay, to further develop your ability of getting good and better memory B cells. Okay, so here is just showing the use of your secondary immune response because of your memory B cell. Okay. So here, finally, we're going to get to those secondary lymphoid tissues. And yes, I know I only have about 10 minutes. Let's get to the things that are really important to know. And this part is easy. Okay, the secondary, the description and function of secondary lymphoid tissues, they're practically the same thing that happens in different places. So if you know what is happening at the lymph node, you're also going to know what is happening at your spleen, okay? And the same type of situation is going to happen at your mouth, the mucose-associated lymphoid tissue. We're going to mainly focus at the lymph node and at the spleen. So here's a picture of your lymph node again, right? So you know that you have a capsule, and coming from like right under the capsule, you are going to have, okay, you're going to have little spaces. So very tough capsule, and then you're going to have spaces right under the capsule. Why? Because the fluid that is coming through those afferent vessels is going to be dumped over there, and it has to have a space to go. You have to have a space that the fluid is going to get inside. So this space here, you call the subcapsular space or subcapsular sinus. Okay? So then you have the outer part, that is the cortex. You have a part close to the cortex that you call it the paracortex. And then you have the center they call the medulla. And inside the medulla, you're going to have more spaces for flow of the fluid. So you call those medullary sinuses. They're not showing here, but we're going to get to that. Okay. So fluid can get from the lymphatics, and cells are going to get to the lymph node from lymphatics. But you also have here those arteries, right? They're bringing circulation to the lymph node. So you have here your lymphoid artery, and here you have lymphoid vein that comes out, right? But the idea here is when you get to that capillary bed, this is where you're going to find your specialized tall endothelial venules, right? The cells that are called high endothelial venules. Because remember, those cells are going to allow passage of your lymphocytes and other cells to get to the lymph nodes, okay? So inside of your lymph node, you're going to have structural cells. You call those cells stromal cells, okay? So structural cells are stromal cells. And then you're going to have some other cells that are part of the functional part of the organ. So those functional cells, you call them parenchymal cells. So if it is structural, it's stromal, at least it starts the same way. Structural, stromal. Then functional is, oh well, parenchymal. I would like to call it functionalimal, but that's not how it is. Okay, call them parenchymal. Okay. All right, so here taking a bit of more amplified view on those structural cells, okay? You have those little fibers. So those are called reticular fibers, okay? So in these reticular fibers is where you're going to find your cells. They're going to basically be using these fibers like train tracks, so they can navigate around the tissue by moving through those train tracks, okay? So here you see fibers and cells that are associating with it, okay? What is important here, okay? Line, fibers, conjugate system, okay, so that you can read on your own. And okay, finally you get another point here. Question is, which of the following is the main site of activation of your adaptive immune response? That's an easy question. A. Okay. All right. So here, structure of a lymph node. You have the cortex. You have things close to the cortex. There's paracortex and medulla. At the cortex, you're going to find the B cells. At the paracortex, you're going to find your T cells. So they're going to be circulating around there. In the medulla, you're going to find a lot of spaces for fluid flow. And then the fluid flow is going to go through your efferent vessel. All right. So more on this structure here. The capsule is a tough thing that surrounds the lymphoid, the lymph node. protruding here from the capsule, you're going to find those little fibers that they subdivide the cortex into these different rooms. Okay, so those fibers here, you call them trabecula. So you have the capsule and trabecula, and then you look between two trabecula, you get basically like a pizza slice. Okay, so you have several pizza slices of rooms, different rooms that are inside of your lymph node. Okay, trabecula. All right. We talk about subcapsular sinus and about the medullary sinus. Okay, we have it in there. Okay. We talk about where the B cells are, and they are in your follicles in the cortex. So once the B cells sees the antigen, then you start getting your germinal center. Okay. All right, translation for all this stuff. The respective cells get to their zone and they get activated or not, but if they get activated, they have to get out of their zones and go circulate and find the trouble. Okay. All right, so that is as much as I'm going to talk about the lymph node. So here we're going to go into the spleen. Okay, so what am I going to do here? I'm going to put the spleen session in the video because the function of the spleen is very similar to the function of the lymph node, okay? And it's very easy to follow. So I'm going to add that to the video. So I'll hold you responsible to understand what's going on with the spleen, okay? So when we meet on Tuesday, I'll just flight you through important issues, and if you have questions, bring the questions on Tuesday about the spleen, okay? And then we get to the next topic. All right, so looking here at the spleen, it's right there close to your stomach, okay? So the spleen is right here, and it is your largest lymphoid organ, right? So what is happening here with the spleen, you're gonna have this big artery, the red one, okay, splenic artery that goes in there, and coming out of there, you're gonna have this splenic vein. Now, the way that this thing is working inside the spleen is a lot different than it's working everywhere else in the body, okay? Now, the place that you have both of the artery and the vein that are entering or leaving the spleen, this part here you call the hilum, okay? So this, like bottom part of the kidney bean shape, this is the hilum. Okay. Now, what is the spleen good for? It's going to filtrate the blood, so it's going to look for parasites in the blood, but that's not the only job of the spleen. It is also going to store platelets, okay? So it's a source of platelets, and is also going to get rid of damaged blood cells, red blood cells, or tagged red blood cells, opsonized red blood cells. It's going to get rid of those. And you're going to see here how it's going to happen. Okay. So areas of the spleen. You have a very strong capsule, okay? And also from the capsule, you have the trabecular, just like you had with your lymph node, okay? So you have the subdivisions. And then you have a different type of organization here, okay? So before I go into organization, I'm just making sure I cover this here. Okay, damaged red blood cells, they are disposed in the spleen. If you have bacteria or viruses in the blood, they get trapped in the spleen. If for some reason you don't have your spleen, then you're more susceptible to infections, like bacterial or virus infections, right? Although the rest of your immune system and your lymph nodes should take care of that, but you are more susceptible to infections. All right. So besides of that, you can reserve some blood in the spleen just in case you need it when you're exercising very vigorously or you need some blood for any other reason. You're going to store also platelets and some monocytes in case you have some infection and you have to liberate monocytes. Okay. So areas of the spleen here. So defense against blood pathogen. So you have mainly two areas in the spleen. You have the red pulp and you have the white pulp. Okay, we're going to look into those areas here. Okay, so let's see. The artery that goes into the spleen, that is your main artery right there, called the trabecular artery. And then it's going to be subdivided here into a central arteriole. So artery is a big one. When it branches out and becomes smaller, it's called arteriole, right? And then the arteriole is going to branch again into follicular arterioles. So central arteriole goes in the middle. Follicular arterioles are branching out from that one. Okay. In the center here, this is where you're going to have your white pulp. Yeah, here is darker, but that's where you're going to have your white pulp. And on this side, you're going to have your red pulp. You have better pictures of it. So let me go to the following one. Okay, I like this video, so I put the link here so you can go back and watch the video. Really, really, really informative. All right, so you have the capsule, you have trabecules, and then here's your splenic artery, okay, central arteriole, okay. Now, here, you're going to see this sheath, okay. I know I have a problem pronouncing that, but here you have a lymphatic sheath, and here is where your T cells like to hang around, and also some macrophages, okay. Now, when you look here, you're going to see that the lymphatic sheath is where you're going to have your white pulp, okay? So this is going to be close to, like stuck to your arterioles here. This is where you're going to have your white pulp. Around that is where you're going to have your red pulp, and there is a reason for that. Okay, so what is happening here? You have the periarterial, you have the sheath that is around there, okay? So let me see here. This area here, you're going to find the T cells, and then close, like on the border, close to your red pulp, you're going to find those follicles. Those areas is where your B cells like to hang around, okay? And it's not very much at the red pulp, but it's very close to it. Is vicinal, or is very very close to your PALS area, periarterial sheath, because this is where the T cells are, and then this is where the B cells are. So you're going to have a place where they can interact with each other, okay? Now, at the end of those arterioles, or these little capillaries, you're going to find that you have open-ended capillaries, meaning that they are not going to be connected to the veins. When you look at the circulation, the blood here is not in a closed circulation. This is more like an open circulation. So the blood is going to spill out and leak out of those arteries and flood all of the zone. And this is why it's called the red zone. Okay, so let's see what happens here. You see, this is your artery, and at the end of the artery is going to be open-ended, and the blood is going to spill out. Okay, when the blood spills out, is going to be filtered over here, is going to interact with macrophages and other stuff, and then if the red blood cells are old or not functional or not bending well or tagged, they're going to be killed by macrophages. If the blood cell is healthy and flexible, it can squeeze through little openings and get back to the venous circulation, to the venous side. So the vein is going to be also part of this filter system, okay? So let me see here. All right, so here you have your white pulp, all right, the white pulp in the center. At the very edge of the white pulp is the margin of the white pulp, so you call that the marginal zone, okay? Here you have your periarteriolar lymphoid sheath, you call that PALS. This is where your T cells hang around. Here you have your follicles, that's where your B cells hang around. And in this outer area, you have your macrophages that are going to clean up the blood. Okay. All right, so let me see what I remember. I still have a couple of minutes. I will take my minutes. All right, so here, where the blood spills out of your arteries and before it gets to the vein is going to find a lot of filtrating type of structures, okay? And those you call the cords, okay? So here in this mesh, you're going to find a lot of macrophages that are hanging around in these cords, and they are ready to pick up damaged cells or viruses or stuff that can be in the blood, okay? So the red pulp is really what mainly makes the spleen. So you look here, like 75% of the spleen is made up of red pulp. So what is the percentage for your white pulp? Everybody awake? About 25 is right there, even if you're not awake. Okay. So a lot of red pulp. So what is happening here in the white pulp? Okay. So in the white pulp, you're going to have dendritic cells that are going to hang around, okay? If they catch something, they're going to go and present to the T cells. The T cells are going to be around here. T cells are here, right? Dendritic cells are hanging around. So they find something, they present to the T cells. T cells, if they get activated, they're going to go and interact with the B cells that are in the follicle. But at the same time, you know, the B cells are going to have to see the antigen, so they're going to be antigen experienced. Okay. Let me see what else I did not cover. This is about the same. Okay. So here is also a very nice schematics of what is happening. Here we have your white pulp, right? What kind of cells like to stay around here. Then you have the marginal zone, and you have your red pulp. Okay, and then what kind of cells like to hang around there. And here we have a nicer representation of what happens to the artery. And here is your white pulp, and then it spills out into the red pulp. There is open circulation here, and then it's going to be caught up by the venous side. Besides lymph nodes and spleen, you also have those mucosa-associated lymphoid tissues, and in general, you call those MALT, right? What do? They're going to protect your mucosa. And mucosa, remember, is coming into contact with your outside world, right? So after the immunologists knew about this mucosal tissue, so they start to get more specialized, and they say, oh, look, when you have your nose or your nose and mouth, let's call that nasal associated lymphoid tissue, or NALT. And somebody else said, oh, look, but now here is associated with the bronchus, bronchi. So let's call it bronchus-associated lymphoid tissue, or BOUT. And then somebody else said, okay, now the gut one is really important, so we call that gut-associated lymphoid tissue, or gout. They got to have a sense of humor, right? So now you have mouth, naut, bout, gout, and you can do a little song with it. Okay. So within those mucosal-associated tissues, you have some specialized structures, right? So you have those things that are tonsils and adenoids. They're basically, the same thing, but they do a bit of different function, okay? Like the tonsils, you can actually see the tonsils, okay? You can see your palatine tonsil. Let me see here. Where is the palatine? Right here. So if you look at the palatine tonsil, it is, if you look in the mirror, you're going to see in the back, like by going towards your throat, you're going to see these structures that are on the sides of that. Oh, now I forgot this structure, the name of this little structure. What do you call it? No. Epiglottis is what blocks your airways when you swallow. The sininho, the mouth, the little bell is not a bell. No, the epiglottis is in your throat. Huh? Uvula, there is it. Yeah, uvula. Although it doesn't look like a grape to me, but anyway. So look on the sides of your throat, you're going to see your palatine tonsils. So there are a couple of them. And then if you look really, like, you stick your tongue out and you look all the way in the back of your throat, you're gonna see here your lingual tonsils right there on the back of your throat, okay? And that's also, you know, important for taste. Okay, you have papillary buds close to your tonsils. Okay, and let me see here else. Pharyngeal. Where are the pharyngeal tonsils? So those are palatine, lingual. Okay, so you cannot see the pharyngeal tonsils because they are in your throat. Okay, now when you look here at pharyngeal adenoid. Okay, so here they are interchangeable, but the adenoids, if you look at this, is like all the way between, like after your nose. Okay, you see this is an entrance for your nose and nostril right here. Okay, so all the way in the back. So those adenoids, they are not really connecting with whatever you swallow. Okay, so the adenoids are mainly going to filter the air. So they're going to be trapping stuff that is coming through the air. Now the tonsils are mainly going to trap stuff that you're putting through your mouth. Okay, and it could also be the air, right? Sometimes you breathe through your mouth too. Okay. All right, so I put this slide here to differentiate the tonsils from the adenoids. So here you can, you know, read on your own, and you're going to see basically their functions. So I put some functions here that are more important, right? Adenoids are going to be catching airborne pathogens, and then tonsils are going to be catching whatever is pathogen that comes through your mouth. Okay, so here is a little picture showing your gout. Did I talk about the M cells here before? No, I never mentioned those microfold cells. Okay, so when we get to the mucosal immunology, I'm going to mention a bit more about these M cells, but they're really, really interesting because they are on those, like sitting on those Peyer's patches. Okay, so if you look in the intestine, you're going to see that your the villi and the microvilli, they have little areas in which they have this concentration of immune cells, right? It's just like the lymph node, but it's not encapsulated. Okay, so it looks like a lymph node, but it doesn't have a capsule. This is a main difference between Peyer's patch and lymph nodes. Okay, so what happens here, like at the very top, right at the surface, close here to those microvilli, you're going to have some very specialized cells that do not have microvilli, which is actually not showing here, but those cells, they are very important at allowing samples of whatever is inside the lumen of your intestine, you know, to be shown to the other side. And those cells are really cool because when you have friendly bacteria, those cells are important to teach your immune system not to attack the friendly bacteria. And when you have pathogens, then those cells are going to be important to bring the pathogens to give it to dendritic cells that are going to, you know, process the pathogen or take it to the Peyer's patch and activate some B and T cells. Okay, so it's a lot of things are happening in the Peyer's patch that is very similar to what happens at the lymph node. Okay, but those microfold cells are really cool cells of Peyer's patches. Okay. Oh, so here I have more on that. So here is a microfold cell that doesn't have those microvilli. Okay. So the other cells in the intestine, they do. So these guys are kind of bald. Okay. So they help samples or antigens that are in the gut to come across without being destroyed. Okay. So they come across without being destroyed, and they are presented to immune cells, mainly to innate immune cells, like you see here, macrophage and dendritic cells. And in case they find pathogens, those cells are going to go here in other areas of the Peyer's patch. In the center here, you're going to find B cells. Close to the B cells, you're going to have the T cells. So the same type of arrangement as you find in your lymph node. So in case they find pathogens, they're going to come and activate the T cells. T cells are going to interact with the B cells, and then you're going to get activated lymphoid cells, right? Lymphatic cells here. And those lymphocytes are going to come out of the Peyer's patch, and they're going to go to the nearest lymph node. So the lymph nodes, they're responsible for surveilling the mucosal tissue. Those are called mesenteric lymph nodes. Okay, so you have seen that before. It comes out from the Peyer's patch, go to mesenteric lymph nodes, and then you have further activation and proliferation of your lymphocytes.